हेलो स्टूडेंट्स, वेलकम बैक। एनईपी के फोर्थ सेमेस्टर जोलॉजी के बच्चे जो इस बार एग्जाम्स देंगे, तो आज हम यूनिट 2 के 1 मार्क इंपॉर्टेंट क्वेश्चंस डिस्कस करते हैं। सो न्यूट्रिशन चैप्टर में से क्वेश्चन नंबर 1, व्हाट इज ग्लूकोनियोजेनेसिस। ठीक है, तो इट इज सिंथेसिस ऑफ ग्लूकोज। कहाँ पे होगा? लिवर सेल्स में होगा। नियोजेनेसिस मतलब हम ग्लूकोज को जनरेट कर रहे हैं बट नए प्रोडक्ट से अदर देन कार्बोहाइड्रेट। सो इट इज बेसिकली सिंथेसिस ऑफ ग्लूकोज in liver cells from non-sugar sources like fats and amino acids. So that is called as gluconeogenesis, right? Now, what are essential amino acids? These are those amino acids which cannot be synthesized inside the body. तो कहाँ से requirement पूरी होगी? Of course, from the diet. ये 8 एसेंशियल अमीनो एसिड्स हैं, नेमली आइसोल्यूसिन, ल्यूसिन, लाइसिन, मिथ्योनिन, फेनाइलएलानिन, थ्रियोनिन, ट्रिप्टोफैन एंड वेलिन। ठीक है, इसका मैंने आपको एक की भी दिया हुआ था इन वन ऑफ माय वीडियोस, मैट विल फ्लाई। सो दैट इज हाउ यू कैन रिमेंबर दीज 8 एसेंशियल amino acids. Now name the salts present in the bile juice. ये क्वेश्चन कई बार पूछ लिया जाता है. So there are primarily 3 important salts, जिसमें कि 86% जो predominant salt है, that is sodium bicarbonate, and the other salts are sodium glycolate and sodium taurocolate. Now, what is emulsification of fat? Very important question. It may come for the exam. See, emulsification of fat का मतलब होता है कि आप जो फैट के लार्ज ग्लोब्यूल्स हैं, जो ट्राइग्लिसराइड फॉर्म में जो फैट हमने इनटेक किया, उसको वी ब्रेक डाउन इनटू टाइनी ड्रॉपलेट्स. ठीक है, एंड इन द प्रोसेस इट गेट्स मिक्स्ड विद वाटर बेस्ड लिक्विड्स. ठीक है, so that those small tiny droplets जो आएंगी, they can then be accessed by the digestive juice and the enzymes in the digestive juice. So this function of emulsification of fat, सीधी भाषा में कहूं तो बड़े ग्लोब्यूल को छोटे ग्लोब्यूल में ब्रेक करना. कैसे होता है ये? तो this is done by the salts that is present in the bile juice. So the bile in the body acts as an emulsifier to increase the surface area, and that helps the enzymes to break down these fat molecules. Now, what is zymogens? ये भी important है exam से. These are inactive forms of enzymes. Precisely which enzymes? The protein digesting enzymes. So these are inactive form of enzymes, mainly peptidases, that prevent premature enzyme activity, protecting the cell from self-digestion. What is lactyle? Another very, very important question, most likely to come for exam. So lacteals, they are specialized lymphatic capillaries which are located at the center of each villus in the small intestine. ठीक है, और इनका फंक्शन क्या होता है कि ये एब्जॉर्ब करते हैं डाइटरी लिपिड्स एंड फैट सॉल्युबल विटामिन्स which otherwise are too large to be directly absorbed into the blood. So they get into the blood through these lacteals. Now, how is fructose absorbed in the enterocyte? So it is absorbed by facilitated diffusion. Next question, what is lactose intolerance? So this is a common digestive condition where in the small intestine, the lactase enzyme is not produced in the sufficient amount. So it doesn't break lactose into its corresponding monosaccharide units, galactose and glucose. So what it causes is lactose रह जाता है small intestine में, and then it is decayed by the bacteria that is there in the colon. Right, so it will cause symptoms like bloating, diarrhea, gas, and abdominal contents, cramps within 30 minutes to 2 hours of consuming the dairy products. Now, what are chylomicrons? Another important question for exam. So chylomicrons are large lipoprotein particles. ठीक है, lipids conjugated with proteins. ठीक है, lipid molecules coated with proteins. So these are large lipoprotein particles that transport the dietary fats, precisely triglycerides and cholesterol, from the intestine, from the enterocyte to the bloodstream. So now composition wise, they are composed of around 90% of triglycerides along with some phospholipids, cholesterol, and the protein is apolipoprotein B. Now, another important question, what is the function of micelles in fat digestion? So micelles क्या है? These are spherical complexes which are formed of bile salts and phospholipids, ठीक है, that digest fat products, fatty acid, monoglycerides and cholesterol, and make it soluble into the aqueous environment. ठीक है, and this helps the easy absorption into the intestinal cell. So micelles are basically spherical complexes of bile salts and phospholipids that transport digested fat products, fatty acids, monoglyceride, cholesterol, through the aqueous environment of the small intestine into the enterocytes for absorption. So they increase the solubility of fats, enabling the absorption. Now, how is the intestine adapted for absorption? See, the intestine, it has a massive surface area, and this surface area is because of its very long length. The small intestine is about 6 meters in human beings. And further to increase the surface area, it is provided by finger-like projections called villi. And villi, they further have fine threads, microvilli, which both of these, they increase the surface area, right, for absorption. Then across these walls, rapid diffusion occurs. Okay, all the villi and microvilli, they are richly supplied by blood capillaries, and in the middle of the villi there is a lectile to quickly absorb nutrients. Further, the movement in the intestine, that is the peristaltic movement, it helps in the maximum absorption. So the total length of the intestine being subjected into folds, microvilli and villi, plus easy accessibility, rich supply of blood, okay, via the blood capillaries and lectines, and the muscular contraction and relaxation movements throughout the length of the intestine that ensures maximum absorption. Next question from respiratory chapter now. What are respiratory substrates? So the compounds that are oxidized in respiration to release energy, they are called as respiratory substrates. Right? So now what is respiratory surface? वो surface जिस पे exchange of gases होगा. So that is called as a respiratory surface. Now, these following questions from 3rd to 8th, these are very important. इनमें से कोई ना कोई हमेशा एग्जाम में आता है. What is tidal volume? कई बार पूछा गया. It is the volume of air inspired or expired with each normal breath. जब हम resting position पे हैं, जब हम कोई activity नहीं कर रहे, उस time पे हम जो normal inhalation और exhalation कर रहे हैं, that is called as the tidal volume, and it amounts to about 500 ml in the average adult man. इसमें से 350 ml alveoli तक चली जाती है, और बाकी की जो 150 ml है वो रेस्पिरेटरी ट्रैक्ट में एज अ डेड स्पेस वॉल्यूम constitute करती है. Now, what is inspiratory reserve volume, IRV? It is the extra volume of air which can be inspired by forced inhalation after the normal tidal volume. ने normal inhalation किया, उसके बाद जो forceful आप extra amount of air inhale करेंगे, that is called as inspiratory reserve volume, which is about 2000 to 3000 ml. Likewise, expiratory reserve volume इसका उल्टा हो गया. The extra volume of air which we expire by forced exhalation after a normal exhalation. एक बार normally exhale out किया, उसके बाद जो forceful हम exhalation करेंगे, ठीक है? So that is called as the expiratory reserve volume, which is about 1000 to 1500 ml. What is the vital capacity of lung? So the vital capacity is the maximum amount of air that can be expelled from the lungs by forceful exhalation after a forced inhalation. मैं लंबा सांस खींचूंगी और उसके बाद उसको छोड़ूंगी इस लंबे सांस को, तो that is the maximum amount of air that can enter into the lungs. So this is basically sum of TV, the tidal volume, IRV and ERV, and which is around 3.5 to 4.5 liters in a normal adult person. What is the residual volume? The volume of air which stays back in the lung alveoli after the forceful expiration. जब हम exhale out करते हैं, चाहे हम forcefully exhale करेंगे, कभी भी alveoli पूरी तरह से empty नहीं होते. अगर हो गए तो उनकी walls एक दूसरे के साथ stick कर जाएंगी और alveoli collapse कर जाएंगे. So this is a body adaptation कि उनके अंदर कुछ वॉल्यूम ऑफ एयर हमेशा रहती है even after forceful expiration, and this is called as a residual volume, which is about 1500 ml in all the alveoli taken together. Now, what is the total lung capacity? So it is the amount of air in the lungs and the respiratory passage. ठीक है, after a maximum inhalation लंग्स में सारी एल्वियोलाई में जितनी एयर पहुंचेगी और जो रेस्पिरेटरी ट्रैक्ट में जो एयर ट्रैप्ड रह जाएगी, अगर आप उन दोनों को सम करोगे, सो इट इज बेसिकली द सम ऑफ वाइटल कैपेसिटी, ठीक है, which was 3.5 to 4.5 liters, and the residual volume, that is 1500 ml. So if you add both the things, it comes out to 5 to 6 liters, which is the total lung capacity. Next, what keeps alveoli from collapsing when they are empty? एक तो क्या है कि कभी भी empty नहीं होते, there is always a residual volume of air. In addition to this, the lung alveoli, they are lined by a thin film of phospholipid, which is lecithin. And this phospholipid layer, it acts as a surfactant, which keeps the, तो surfactant का function क्या है? ये actually जो alveoli की wall है, उसको moist रखता है, and it lowers down the surface tension, which prevents it from collapsing. Now, what is Herring-Brewer reflex? Now, this is another protective mechanism of the body to prevent excessive inflation of lungs, alveoli, during conditions of excessive breathing. जब हम heavy exercises करते हैं, है ना, weights उठाते हैं, तो the body requires extra amount of oxygen. So, कि एक अपनी कैपेसिटी से जरूरत से ज्यादा लंग्स इन्फ्लेट ना हो जाए, नहीं तो वो बर्स्ट हो जाएंगे, ठीक है। मैकेनिज्म है, दैट इज द प्रोटेक्टिव मैकेनिज्म इज द